When we met last, we had met from the ranch where we were talking about different types of bacteria that could cause mastitis in cattle. And one of the groups that we talked about were the gram-negative Enterobacteriaceae, a family of gram-negatives that it's hard to differentiate amongst. One of the ways that we talked about differentiating between them was to look at the way in which they ferment glucose. So as you are working to identify unknowns, once you've found out that an unknown does ferment glucose, it turns the tube yellow in a phenol red simple glucose fermentation experiment, then we moved on to looking at the use of the MRVP test to tell us what pathway an organism uses in order to ferment glucose. There are two pathways. There's the mixed acid pathway and the 2,3-butanediol pathway. So this test requires you to take your purple top tube, take out a certain volume, a small volume, put it into a new tube, and then add the pH indicator methyl red to that little volume. And so upon doing that, you will see that the tube either turns red, and we recognize that red color as indicating a positive result for the mixed acid fermentation pathway. So this table is simply reviewing that. Yellow means that it is negative for the mixed acid fermentation pathway. Enough acids were not produced to overcome the buffer that's present in that tube, and therefore it stayed a more neutral to alkaline pH, whereas this one is more acidic. It overcame the buffer, and we can see that turning red based upon the addition of that methyl red pH indicator. Now, the other part of the test is the more complicated one, because recall that with the VP part of the test, the remainder of your purple tube's volume, to that we added two reagents. You'll add the alpha naphthol and the KOH, and look for the reaction to take place over the course of 40 minutes. It takes about 40 minutes or more to get to the point where the tubes are stably pink or stably kind of brownish yellow. Negative is this brownish yellow color, and we see that that's an indicator that the 2,3-butanediol pathway was not utilized to ferment glucose. That is, there's no acetoin, which is the precursor to 2,3-butanediol, to react with the alpha naphthol and potassium hydroxide. Whereas in tube A, there was that acetoin present to cause that positive reaction to take place. That being said, we have a little review visual to help us recall all that's going on in this test. Notice that glucose can get fermented using the mixed acid fermentation pathway, tested for in the MR part of the test, indicating stable production of lactic, succinic, formic acids. Whereas if we look to the VP part of the test, the indicator that 2,3-butanediol has been formed, then we're able to see the stable production of acetoin, which is detected by the addition of alpha naphthol and potassium hydroxide. One of two possible fermentation pathways for glucose. Now, one thing to bear in mind is that unless you've got contamination in that tube, then you shouldn't see that your organism is positive for both of these pathways. Of course, we may see that it's negative for both, because of course you may have a glucose non-fermenter, such as Pseudomonas aeruginosa, that doesn't ferment glucose and therefore uses neither pathway. E. coli is going to be MR positive, so we should see the red color, and that may be a confirming test for your Escherichia coli. Or if you think you potentially have the Enterobacter aerogenes environmental Enterobacteriaceae, you might see an MR negative where the VP, it's going to be really exciting, right? It begins to turn red at the top of the tube after about 20 to 30 minutes, and then the whole tube starts to turn closer to 40 minutes. That was a quick review of the MRVP as we covered it last time, and so we need to launch into some new tests now, beginning with the Simmons citrate auger. This is yet another test that helps us amongst that family, Enterobacteriaceae, wanting to be able to differentiate things like E. coli from things like Proteus mirabilis or Enterobacter aerogenes or Klebsiella pneumoniae. These are all members of the family Enterobacteriaceae. And this is a great test to help us do that. We're looking for the presence of an enzyme called citrase that can break down citrate into oxaloacetate and acetic acid. And so a recognition of the fact that these are familiar intermediates to us from the TCA cycle. So remember that citrate is the first intermediate in the TCA and oxaloacetate the last. So recognizing that we're looking for this reaction catalyzed by citrase, the oxaloacetate is hydrolyzed into pyruvate and CO2 in the final stages of this enzyme catalyzed reaction. And the presence of CO2 leads to the production within this medium of a very alkaline compound, sodium carbonate. Sodium carbonate may ring a bell to you. It's a water softener. It is alkaline though, and it turns the pH to a more basic pH, which can be seen based upon the pH indicator that we put into this auger is called bromothiamyl blue, and the bromothiamyl blue turns from green to blue. And in fact, it's not just any blue. It's a beautiful Prussian blue, and we can see it here in this image where the citrate positive is on the right and the citrate negative is on the left. Now this can be very distinguishing, as I said, amongst these different types of Enterobacteriaceae or members of Enterobacteriaceae. Klebsiella pneumoniae and Proteus mirabilis will become this beautiful Prussian blue, whereas E. coli and Shigella dysenteriae, they're going to stay the green kind of nondescript color that we began with when we inoculated the two. So this is a fun one to interpret, and hopefully you have fun interpreting that on your unknown today. Let's move along looking at the next test, which is for actually gram-positive rods. And we've got two in a row that are going to help us to distinguish amongst the gram-positive rods. Let's begin with the spirit blue, or looking at the ability of an organism to hydrolyze triacyl glycerols, cleaving them at the ester linkage that links between the glycerol backbone and the three long fatty acyl side chains. So lipase is the enzyme that does that. And the truth of the matter is that the reason that bacteria have to secrete lipase into the space surrounding the cell is because it is just triacyl glycerols are just too big to get taken into the cell in their fully esterified form. And so instead, the bacteria have to secrete the lipase to break these into smaller units so that they're small enough for the bacterium to take up. Right? Size does matter, doesn't it? And so we recognize that glycerol, the backbone, that can enter into catabolism at all of you remember as glyceraldehyde 3-phosphate, the intermediates in glycolysis that are in equilibrium with one another, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. It enters at that first stage of the trio stages, or the 3 carbon stages of glycolysis. Now, sometimes it is catabolized and sometimes it's used as a precursor metabolite in an anabolism. So, we can look at that in either way. The fatty acyl side chains, why they're broken down 2 carbons at a time. And remember that that forms acetyl-CoA. And that acetyl-CoA enters at the top of the TCA cycle where it runs that TCA cycle. So lots of energy derives from chowing down on a fat. These bacteria that can secrete lipase and use fats as a fuel source keep their TCA running, right? They keep it moving and on the move with all of those acetyl-CoA molecules that are entering at the top of the TCA and turning the TCA. Now, of course, it must be that spirit blue auger contains oil, and explicitly it contains olive oil. The olive oil is bound up within the auger, and when the bacterium secretes lipase, it will use up the olive oil in the area surrounding the bacterial growth. And so there will be this halo of clarity. I wish I had one of those, a halo of clarity, around the bacterial growth showing a positive. This is a sweet picture of a lipase positive when held up to the window so that you can see the light through that clearing area around the growth. Please remember that this plate was let to sit for quite a long time so that it had been able to take in a lot of the fats that surround the growth. And therefore, the area of clarity is very evident. You may have a little harder time. Make sure you hold it up to the light and make sure you ask your TAs if you're uncertain as to whether or not your organism is lipase positive. Now, a lot of bacteria that can secrete lipase can also secrete the enzymes that break down large starches. So things like amylose and amyliopectin, the branched polysaccharide, they can secrete the enzymes that break the glycosidic linkages in the starches. So amylose and amyloidopectin are hydrolyzed by some bacteria. They are hydrolyzed using two enzymes. One is called alpha amylase, and the other is called oligo-1,6-glucosidase. These two enzymes, respectively, cleave the 1,4 and the 1,6 linkages within a starch. Let's take a quick look at that picture and come back to the rest of this text in a moment. Notice here is a cartoon depiction of a small snapshot of a starch where you can see that alpha amylase is cleaving the 1,4 linkages, those that link together carbons 1 and 4, and oligo-1,6-glucosidase is cleaving the 1,6 linkages. Now, once again, this is a size matters argument. Organisms that secrete these enzymes can bring in the subunits of the starch because the starch itself is too large and cumbersome to bring in and pass through the cell in its large size. So organisms like Clostridium and Bacillus, they are differentiated from one another using this test. Bacillus is positive for starch hydrolysis, whereas Clostridium is generally not. So it helps us differentiate. The other thing to be said about this is that your unknowns, some of you have Mycobacterium smegmatis. It's negative for hydrolysis of starch, whereas if you have Bacillus subtilis, it is positive for hydrolysis of starch. So a great way to differentiate amongst our gram-positive rods. Now, what happens in this auger is that there is a lot of starch, obviously, but there's also, we have to have a color indicator of some sort so that we can see the halo in the region where these organisms have secreted the enzyme to degrade the starches. In order to create that halo, we put iodine onto the auger. So after you have grown up your organism on the starch plate, you will saturate it with iodine. You will take the grams iodine, drip it on, and please don't make a swimming pool of iodine. It's not necessary, but do cover the surface entirely. Let it sit, be patient, wait for maybe 30 or 40 minutes until the iodine has completely soaked in. And then either put your plate onto the overhead so that you can see the halo or hold it up to the light, and you'll begin to notice that if it's amylase positive, there's this beautiful light, almost yellowy brown halo that surrounds the bacteria where they have secreted the enzymes to break down the starches to bring in the sugars as the energy sources that they want. So this is a beautiful example of an amylase positive versus an amylase negative organism. Writing down what you see there, there's a nice clear or kind of yellowy zone or halo around a positive, such as Bacillus subtilis. Whereas if the organism is negative, typically you just see iodine everywhere and there isn't the evidence of that beautiful halo. So these gram-positive rods, the ability to break down macromolecules by secreting enzymes into the space exterior to the cell is very important in helping us to identify and differentiate amongst them. Now today is a very exciting day because you are needing to determine the unknown identity, and so you need to today do any other tests that you maybe had to go back to because for one reason or another you didn't get them done. You are going to want to confirm the identity of your unknown with your instructor or your TA. Please, before you go to that person, treat them with the utmost respect by ensuring that you're pretty sure of what you have, not based on your hypothesis, but based upon the definitive tests of your biochemical tests that you have performed on your organisms. Please do get your report done. Your TAs will specifically instruct you as to the due date for that report. Please remember that you can use your late coupon. But if you don't use your late coupon, remember that we do deduct for every day that it is late. So please look to your TAs to give you specifics on all of the due dates. And I hope you are so jazzed to ID your unknown today and rock your TA's worlds.